Try again. I've got somebody at the door.
Okay,
We're live now.
we're live.
Hello and welcome to Tensegrity Party 2.3. My name is Graham Scarr from Nottingham, UK.
And I'm Chris Morita Clancy from just outside of Vancouver in Canada.
And I'm Susan Lowell de Solorzano in Washington, D.C.
And it is our intention to promote and share the biotensegrity concept with as many people as possible and to include you all in that mission. The Biotensegrity Party is an all-volunteer production of the Stephen M. Levin Biotensegrity Archive, a not-for-profit organisation whose mission is to educate, raise public awareness and build community in the field of biotensegrity, and to foster and forward discovery, research and understanding in the fields of science, health and medicine. And today we have our guests Gerald Pollack from the University of Washington in Seattle and Niall Galloway from Atlanta, Georgia. But first, let's introduce the team. Mariana Barreto from Mississauga, Ontario. Hello, Mariana.
Hello, everyone.
Rachel Tudor from Turnwater, Washington.
But Rachel can't be here today, but she'll be
Oh,
back
right.
in a few weeks.
My pardon. And Lisa Babiot from St. Albert, Alberta.
Hi, everybody.
And Gregory Schudy from, I don't know where he's from actually. Where are you from, Gregory?
Lancaster, Pennsylvania.
Thanks. So Neil, I wonder if you'd open with the toast for us.
Thank you, Graham. It would be my pleasure. It's time to raise a glass. Research and discovery is sometimes about seeing something everybody else has seen and thinking what nobody else has thought. That was something that Albert Szent-Györgyi, who won the Nobel Prize in 1937 for his work in vitamin C. He's said to have said that. And we're delighted today that we're going to have Gerald Pollack, who is one of those men who's capable of thinking something that nobody else has thought. Well, let us all be well and stay well forever. Cheers.
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 Forever.
Cheers.
Thank you, Neil. And next we'll go to our sponsors, which includes Handspring Publishing in East Lothian, Scotland, who have a wide range of authors and books available for movement and manual therapy professionals, and are offering a discount coupon to those on the Biotensegrity Archive mailing list. Handspring also produce a regular newsletter, which is free, and you can join from their website, which provides details of their ongoing webinar series with Elizabeth Larkham. Embodied Biotensegrity with Chris Clancy in Vancouver, British Columbia, who provides an excellent resource for those of you looking to learn more about biotensegrity. Chris is currently offering a 14-day free trial for her ongoing course, Biotensegrity Trailblazers, so check out the chat box for more information. Next, we have Artifact Pro in Madrid, Spain, where you can find wonderfully constructed tensegrity models who also offer a discount coupon to our Zoom participants. Then Integrated Biotensegrity in Alberta, Canada, who are holding a 3-day movement-focused event with keynote speakers John Sharkey, Joanne Avison, Wilbur Kelsick, and other international presenters who you'll be familiar with from the Biotensegrity Party. And there is also a discount code in the Zoom chat. And the fasciahub.com, who are running a free webinar on February the 24th, that's next Wednesday, with Roberto Ciaf, London-based professor who has created the Sigma Q technology. It has an international reputation in the fields of neurology, motor neuron dysfunction, and Guillain-Barré syndrome. So thank you to our sponsors.
And Graham, we also have Congreso Internacional Biotensegridad, which will be coming up 23rd, 24th, 25th of April.
Thank you.
Yeah, it's going to be in English and Portuguese.
Thank you. Things move so fast around here.
And at this point, before we hand the mic over to Neil and Gerald, can we please shut off our videos for the audience? Neil and Gerald, please leave yours on. And that gives a better view for our YouTube audience. If you're here and watching us from YouTube, please say hello, put your questions into the comments. We'll have somebody monitoring the YouTube channel and transferring that over to the Zoom chat. And if you have questions here on Zoom, put them in the chat as they come up. Susan will be monitoring those questions. And we, of course, can't wait for the question and answer period, which will come after the presentation. Thanks all. I'm very excited.
Thank Chris, and over to you, Neil.
Well, thank you very much. It gives me enormous pleasure to welcome Gerald Pollack to this group. In the past, your ears have been burning as we've been talking about you and your work in your absence. And today we have an opportunity to hear directly from the horse's mouth. I'm not going to introduce Gerald's CV. It would take all afternoon. He is very well positioned on the internet, and anybody who would like to look up his recent works are most welcome to do so. Most recently, we heard from Gerald at the Brazilian Fascia Congress just 2 months ago. And it's with great pleasure that I'd like to invite Gerald to take it from here. Thank you very much.
Well, thank you. Thank you, Neil. Okay, I will do so. I guess I need to share screen. Is that okay? Okay, so having heard no comments otherwise, I think it's working. And so I want to talk about the fourth phase of water. And let's see, I have this problem again. Okay, excuse me, let me escape and let me come back to screen. Okay, and then to try to relate this to fascia. And I've got to admit upfront,
we are not seeing your screen just yet.
I'm sorry, you're not seeing it?
No.
Oh, let
Jerry,
me see.
I think you stopped the share and then you were going to change the
Oh yeah.
format and go back in and share again.
Okay, so I should click share.
Yes.
Okay, and I get to the screen.
Looks great.
Presentation mode.
And I'm going to go to presentation mode.
And there your arrows should be working.
Okay, great. Yeah, okay. So as I said, fascia is not something that we deal with, nor is tensegrity, although it's implicit in what we do. So I want to give you a kind of, shall we say, an overview. I know that I don't have a lot of time to do this, but I think some of the work that I'm going to present on water, I think has application to the field of fascia and tensegrity. And it all starts with water, of course. And you're all familiar with the water molecule with an oxygen, 2 negatives, and hydrogen, each one positive. And we found that when the appropriate kind of energy comes in to the water, and I'll deal with the kind of energy in a moment, what happens is that the molecule splits, as this dashed line shows. It splits into two parts. One of them is the positive hydrogen, and the other is the negative OH minus. Negative charge because the oxygen has 2 minuses, and the hydrogen has one plus. This is very similar to what happens in the first step of photosynthesis, where light comes in and light breaks the water molecule into OH minus and H plus. And what I'm going to show you is that this splitting of water, just like the first step of photosynthesis, is pervasive. It occurs not only in green plants and some bacteria, unicellular organisms, but all over, always, all the time. And what we found is that when this splits, that if you start with water that's sitting next to a material surface, what happens is that the OH minuses gather here into this region, and the positive charges, the H pluses, will gather here in what we call bulk water. And this is what we refer to as fourth phase water, this altered water, because it's sitting next to this material surface, mostly hydrophilic surfaces. So we call it fourth phase, and we call it EZ also for exclusion zone, because it tends to exclude any molecules that you have originally in the water. It just pushes them out this way. And because you've got negative here segregated from positive here, there's potential energy. So this system has potential energy. It's kind of like a battery, because it's a, you might say, water battery, because it's got negative here and positive here, just full and capable of delivering potential energy. And so I mentioned the exclusion, the structure of the EZ, which we've been able to deduce over the years, but I don't have time to, to tell you about all of the rationale and evidence behind it. It's a sheet-like structure. So you start with a hydrophilic material sitting next to water, and out of the water undergoes a transition, the water that's right near the surface. And so the first layer, it's a sheet-like honeycomb layer builds. Then building onto that first layer is a second layer and a third layer and so on. And they just keep building, and they can build up to numerous layers. We found in some experiments up to on the order of 1 million molecular layers. So it's very substantial. It's not just 1 or 2 molecular layers. And if you look at the structure of each layer, it's a honeycomb, as I said, consisting of oxygen, oxygens at the vertices, and the hydrogens in between. And if you were to count the number of oxygens and hydrogens in one unit cell, you'd find it's not H2O anymore. And it can't be H2O because H2O, it has no charge, you see, and we've measured here directly with electrodes. This has negative charge, as I pointed out in the previous slide. And so this couldn't be neutral. It has to be negatively charged. And it turns out if you count in one unit cell the number of contributions from each of these, it turns out it's H3O2. So this EZ water is in effect not H2O, it's H3O2. If this were H4O2, it would be twice this, it would be neutral. This is missing one positive charge and therefore negatively charged. So you can see in this video about the exclusion that I mentioned, this is a very tight structure, and you can imagine trying for a molecule to try to get through here. It's got to be extremely small, and almost nothing fits. So that's the reason for the exclusion, and you can see it here. And in this case, we have a chamber here, and we have a gel at the left side. And on the right side, we had water plus particles that are suspended in the water. And these particles are little spheres that are called microspheres, and they're widely used. And you can see what happens. We start with water and these spheres, and something is growing. This exclusion zone is growing from the boundary and pushing the microspheres away. In this particular case, the area here is on the order of 50 micrometers, but we've had as much as 1 millimeter in certain cases. So it's pretty extensive. And this is, we call this exclusion zone water, or just EZ water. And of course, I always point out it works very well in the US because it's easy to remember. But in Europe and other places, unfortunately, it's EZ. So it doesn't work quite as well. Now, so where does the energy come from to do this? I mentioned that this region is negatively charged. This region is positively charged. Charge is being separated. And it's just like your cell phone battery, you got to charge it, otherwise it doesn't work. And this battery is similar. So where does the energy come from? We struggled, I have to admit, for a few years to figure out because you can't sort of take the chamber and plug it into a receptacle and recharge like your cell phone battery. So where does the light, where does the energy come from? And it turns out the answer is very simple. It's light. And we really hadn't thought of it until a student, an undergraduate student, was doing an experiment in the lab. And then he began doing what he was not supposed to do. So here's the experiment. And just start on the left side here. So this is a hydrophilic material sitting next to water and these microspheres. And you can see that the microspheres are here, but they're excluded from the exclusion zone. And if nothing were present to perturb this system, this boundary between the EZ and ordinary bulk water would be straight, parallel to this one. But the student decided to have some fun. There was a lamp, a gooseneck lamp that was sitting right next to him. And he decided to shine the lamp on the chamber. Very simple experiment, you know, just to see what would happen. And the result was astonishing. So this is real data. And this is obviously the artist drawing, happens to be my son, who's a professional artist. And what happens, you can see, he was shining the lamp on this region of the chamber, and the exclusion zone grew. And it grew quite substantially. And he came to get me. He said, I want to show you something. And I went into the laboratory, and I was astonished. And I said, well, turn off the lamp. What happens? He turned off the lamp, and this reverted back so that within tens of minutes, the boundary here was a straight boundary, just as it had been before he turned on the lamp. So it didn't take a rocket scientist to figure out that, you know, apparently photons from the lamp are responsible for building the exclusion zone, and maybe here too. I mean, one mechanism applying, and you add more of this light, and you get a bigger EZ. So we started then to test to see which wavelengths are the most important, and we found that the most important wavelengths are in the infrared. You know, a lot of people don't know a lot about infrared. Well, what's infrared? We know about visible light, and of course, infrared is the wavelengths that are longer than red. And it turns out that that infrared energy is coming not just from your toaster or your oven, where it gets hot and the glowing coils are said to give off infrared energy, but it's all over the place. If you were to darken the room in which you're sitting and whip out an infrared camera that is with a sensor that is sensitive to infrared instead of visible light, you'd see infrared coming from all over. And because this energy is around, you can't get rid of it unless you go to really extreme measures, which are completely impractical. The energy that's responsible for creating EZ is always present, you see. And so because it's always present, what that means is if you've got the right surface, this hydrophilic material sitting next to water, you will always have some EZ water because the energy to create it is always there, and the surface that's responsible is there as well. Now, if you were to add more infrared with like a heater or something like this, then the EZ would grow. And if you take it away, it will revert back to its default configuration or size. So it's all over the place. And we can actually extract electrical energy from this system. So here you've seen this before. This is EZ water and bulk water, and we put one electrode here connected to a lamp, and voila, it works. I can show you this video. So on the right side, you've got a bunch of chambers, I think a dozen or so. Each chamber contains water, 2 electrodes, and a little piece of naphthalene to act as the hydrophilic surface. And basically those 2 electrodes are sitting, one in the negative, one in the positive. This is a switch. This is a magnifier, which covers the light emitting diode LED behind it. And so the video shows what happens. You turn the switch and voila, the lamp lights. So I'm not suggesting that this is anywhere near solving the problem of getting electricity for nature, but we're working on it. And as many of you know, there's so-called valley of death. You have something that works nicely in the laboratory, and then you try to convert it into something that's for practical use, which we do hope to do. We're not there yet. It takes a while to make it work. But now I think most of you are less interested than in electrical energy for the world, but what happens inside the cell. And so inside the cell, depicted here, you've got solids inside, mostly proteins, about 70%, and nucleic acids and other entities. But the surfaces of these are mainly hydrophilic, not all, but mainly. And next to these hydrophilic surfaces, we expect the water to be EZ water, producing negative charge, as you see here. But this diagram is wrong because it doesn't depict the actual situation, especially the crowdedness inside the cell. That's shown here, and even this is a huge underestimate of how crowded it really is. There's not much room left for water, but whatever room is left is going to be mostly EZ water with negative charge. Actually several groups calculated the distance between neighboring solids on average, and the average distance is equal to something like 7 water molecule diameters. So everything is really close together, which means that essentially all the water is going to be interfacial water or EZ water, and it will have a negative charge. And I know that many of you who are watching will know that when you stick an electrode into a cell, you measure negative charge. You measure electrical potential minus 60 or 70 or 80 millivolts. And the prevailing view is it has something to do with the membrane, with the pumps and the channels and such. But I'd like to suggest that that's not the answer, and the answer is in the water. That because this water has negative charge, and the cell has negative charge, I think, and that's consistent with the observation that if you take a gel instead of a cell, no membrane, no pumps, no channels, you get the same approximately electrical potential. So it's difficult to argue that in one case it comes from the pumps and channels, and in another case it comes from who knows what. I think it comes from the water. So if you think about it, a cell is filled with negative charges. All the positive charges are pushed out. These negative charges repel one another, and all they want to do is get away from one another, and that's potential energy. The cell would be happier if all these negative charges were to disperse. So it's potential energy. And so what's the potential energy used for? And I believe that it's used for, I believe is not the right word. The evidence suggests that it's used to do the work of the cell, which is embodied in the folding of proteins. So the way that works is, in any particular cell, take, for example, a muscle cell, because I worked in that field, but it could be a nerve cell or a secretory cell or whatever. And the way that performs work is that the proteins fold. In the case of, they go from one state to another state. And we have evidence that this is driven by a transition from this EZ water to ordinary water, and then it comes back again to restore the initial condition. And illustrating that is this diagram. So on the left side, here's a protein, and it's surrounded by EZ water. And what happens is when the cell is activated to do whatever the cell does for a living, in muscle cells it's contracting, and secretory cells it's secreting, whatever. And what happens is the water melts into ordinary water, and that allows the protein to fold and do what it's supposed to do. And of course, after it folds, it's got to unfold. So it comes back. And as it comes back, the easy water is then restored. So it's a cycle involving easy water to start with, or structured water, if you will, melting into ordinary water, allowing the cell to do its work, and then coming back again. Now, on the right side is a cell that's not, or a protein that's not functioning properly. It has no EZ, or, this is an exaggeration, limited amount of easy water, let's say. And what happens is that it can't do what it's supposed to do. It's impotent because it doesn't have the easy water that can undergo the melt cycle. So this is an area where the water, the easy water or fourth phase water, is centrally involved in the function of the cell by driving the folding of proteins. And I think some of you know about this book. This was in 2001, and it received mixed reviews. Some reviews were enthusiastic, and others said this is the biggest nonsense that ever appeared on the scientific scene. So, but I got to tell you that among discerning people, they'll pick that book out every time. And in fact, this particular character here is still reading it, so it must be compelling in some way. So anyway, back to the cell. So cell is got potential energy that basically drives work. And if you go through the steps that I've gone through, the first is that light builds EZ, okay? And that gives you the negative charge inside the cell, which gives you potential energy. And the energy is driving the work of the cell, which is actually the folding of the protein. And if you connect the dots, light is driving work and folding, light. And so because of this, if you ask the question, where do we get our energy? Well, of course, you know, eating pizza or whatever these guys are, looks like cheesecake, I'm not sure, but we also get energy from light. And the energy from light is absorbed by the water, builds EZ, and gives us energy. And we have now abundant evidence that this is operative in many cells, including the cardiovascular system, where we demonstrate that, that this energy is actually used for helping the heart to propel blood. It's a propulsion system that's based in the capillaries, and it's run on light. And so other cells do much the same. So we may think that, that we depend on food alone, but in fact, just like plants and some unicellular organisms, we also depend on light. And I think many of you don't know this, but you know, if you stop the heart, going back to the cardiovascular system, if you stop the heart, you'd expect that blood flow ceases, but it doesn't. We have experiments, and we're not the only ones. There are a half dozen different groups that have reported pretty much the same thing over the past 100 years, but who pays attention to it, that if you somehow stop the heart, you expect the blood flow to stop, but it doesn't stop, it keeps going. Not at the same speed, at much reduced speed, but it keeps going. And what we found recently is that if you add light, it goes much faster. So I think this is operating in your body. So I'm not suggesting that we photosynthesize, but I am suggesting that we use light in much the same way as plants, green plants use light. First step is the splitting into negative and positive. But as I suggested earlier, it's more generic. It's not just in plants. Now, what about, how does this relate somehow to fascia? And I would say yes, a little bit indirect, but since the topic is fascia, I wanted to of course, bring forth a few ideas that may be relevant. And the first one is the flow in vessels. And another one is in the lubrication of joints. And the third is what happens when something is injured. So I want to talk briefly about each of these three. The first one about flow in vessels. So we found something amazing for us. And again, this is another undergraduate student who was doing what he wasn't supposed to do. And so we found out that Nafion, material we like to use, not the only one by far, but it comes in hollow tubes, just like a straw. And the student found that if he immersed one of these in water, and he put some little spheres in here so he could track movements. You can't see movements of water very easily unless there are particles that are suspended. I apologize for the noise. Guys come once every 2 weeks to do some gardening, and they've got earplugs. So anyway, the student came running into my office to tell me that the flow goes through the tube ceaselessly. It just keeps going. You know, I was, of course, astonished by this, and that's what led us to study the cardiovascular system, where we think something absolutely similar is occurring. This flow is driven by light, by radiant energy. And we've established, the paper is being submitted very, very soon, we've established that this occurs in capillaries, probably in lymphatics, and in cells. There are all kinds of flows that occur in cells, and also almost certainly around fascia, where the proteins are such that EZ water will build on the surface, and when you have EZ, you'll have flow, just similar to this one. Another one is in the lubrication of joints. To discuss the lubrication of joints, first question is, you know, you want to avoid friction. That's what lubrication does. And how do you avoid friction? And the first question is, well, what creates friction to begin with? What's responsible? And, you know, this is a nice illustration of what happens. You can imagine sandpaper, one piece here, and little pieces of grit sticking up and coming down. And if you shear one of these past the other, it's difficult. Take two pieces of sandpaper and they would hardly move. But if you were to separate them, then there's no problem. They move easily. And I think this principle is actually the same. So illustrated here, once again, here's a rough surface, and here's another rough surface. And if you were to try to shear this one past this one, it'd be difficult to do. However, if these are hydrophilic surfaces, some hydrophilic material, and you have water in between, what happens, as I've shown you earlier, is that EZ will grow here next to this surface, and it'll grow next to this surface. And whenever you have negatively charged EZ, you'll have positively charged either protons or hydronium ions, you know, protons stuck to water, with positive charges here. Now, these positive charges repel one another. If you have enough of them, these surfaces will separate and will stay separated. It's kind of like the same thing that happens in ice skating. You put that blade down, it's exerting pressure on the ice, and there's an area that forms just above the ice, area of EZ, and protons sitting at the top. And if your blade presses down, it doesn't actually touch the ice. What happens is it falls on these positive charges, which repel each other and keep the blade away from the ice. And so the principle is similar here. And so now if you were to rub or shear this one in this direction and this one in this direction, friction would be virtually zero. And I think this is manifested in low friction in joints. So here's an example of a joint. You have bones here, a ligament connecting, and you have the joint capsule that sits here with a membrane that sits around it, keeps it all together intact. And you have cartilage that lines this surface and that lines this surface. And I would suggest to you that the same principle that I've just discussed is applicable here. So if you start with the cartilage, because the cartilage is hydrophilic, charged materials, you're going to build EZ here, and you're going to build EZ here, right next to each one of these surfaces. And this is going to be filled with protons or hydronium ions, and they repel each other. So these surfaces actually never actually touch. And that's confirmed by studies in the field that these two layers don't touch each other, despite enormous weights that can be put on here. And I think the reason is it is positive charges that lie in between. And the last one has to do with swelling induced by injury. So, you know, if you break your arm or your ankle or whatever, it swells pretty quickly. And the question is why it should swell so quickly. And I would like to suggest to you that the reason has to do with something to do with the water. And it sort of reminds me of, you know, the guy who has a hammer, and everything he sees looks like a nail because the only tool he has is a hammer. So I tend to focus on EZ and on the water, just as many of you like to focus on tensegrity. And the two are, I think, intertwined in some way. So you've got EZ buildup next to various protein surfaces inside of organelles. And there's a tendency to swell because the water, EZ water, continues to try to build up to actually many layers. But they can't because cross-links between proteins, like rungs of a ladder, you know, in between these two verticals, tend to restrain these structures from swelling excessively. But now, if you got an injury, that's going to tear the cross-links. And if the cross-links are torn, then the EZ water will build unrestrained, and the tissue will swell. And I think this is maybe related. Example, of course, comes from muscle because I've been there in the past. So this is a single myofibril. It's a couple of micrometers in diameter from honeybee flight muscle. Here's one, here's another, and you got mitochondria sitting there all over, and cross-links connecting this myofibril with this myofibril. So the myofibrils, individual myofibrils, can't really expand too much because the cross-links prevent that. But the same thing is occurring even inside the myofibril. And I'll show you a few slides about that because I think many of you know about muscles. So this is an electron micrograph taken of muscles, and you can see these are the thick filaments that run here. And you'd think that these are the thin filaments because they're thinner, but they're not. They're the connecting or tightened filaments that run from the thick filament to the end of the sarcomere, to the Z line. And this sarcomere has been really greatly stretched out to get the actin filaments or thin filaments out of the lattice because they obscure what you want to see. So here you have thick filament, another thick filament, another thick filament, and this is the center of the sarcomere. And if you look at the cross bridges, I don't know what you see, but what I see are simple links. And if you were to turn this vertically, they would look like rungs of a ladder that you could climb. Very, very simple kinds of structure. And you can see this basically all over, especially when the filaments are separated enough that you can see them. This is a kind of surprise, right? Because most of us think about muscle structure as being different. This is another example. You can see them again, here, here. And if you take a cross section through here, you can see these radial linkages that occur. So we also looked at a completely different method to prepare this, and that is the freeze fracture method. You can see structures, but they're not as clear, but they don't involve fixatives and other artificial agents. And this is, you can see this here. So this is the Z line. I'm sorry, it looks like an N because it's turned over. Here are the connecting filaments again, and the thick filaments. And you can, again, see the connecting links. This is a completely different method, but the results are the same. So it looks like the real structure, not the structure that you find in the textbooks, the evidence suggests that the real structure looks like this, with a thick filament here, a thick filament here, and crosslinks between them. And because the myosin molecule looks like this, our best guess is that these structures consist of one myosin molecule from one thick filament, another one from another thick filament, joining to form a permanent crosslink like that. Of course, this model, which comes directly from the structure that you see, not from the artist's conception, differs from what we think of the way muscles work, which is kind of diagrammed here. You know, the idea you've got cross bridges that stick out, and the prevailing theory is that they undergo a rotational cycle, and then they detach, and they do it again and again and again, and all of these cross bridges are then driving the actin filament to move in this direction. The previous slide shows that this is not likely to happen because the structure that is not compatible with this. There are many other reasons to think that that this model fails in so many ways, but that's not the purpose of my presentation. So getting back to what seems to be the real structure, this is a thick filament. And so are these thick filaments, and the thick filaments are held together by these crosslinks, myosin crosslinks. And the actin filament fits into this triangle, into this lattice somewhere. And as I said before, the EZ water tends to build around here, layer by layer, extending out. And ordinarily, they want to keep building, but they can't keep building because they're restrained by these links that prevent these thick filaments from separating more than a certain amount. And so when you think about the water, this could be, let's say, one of those filaments, a myosin or actin filament. It could also be a myofibril or a protein or fascia. And you got a certain amount of EZ water. And the EZ water with ambient energy tends to build. There's always a tendency for this to want to grow. In this case, it's restrained by these links, which prevent excessive growth of EZ from these and separation of these. However, if you're injured, then these are missing. They're broken. And if they're broken, it means that the EZ layers can grow with impunity. They just keep growing. And therefore, these are going to be separated. And if they're separated, there's no restraint to swelling. And then quickly you wind up with something like this, which I did witness myself after a tennis match once on my own ankle. So at any rate, I think that that EZ might help to explain issues that are somehow related to, or at least indirectly related to fascia. That is, you can get flow inside vessels and flow alongside any kinds of hydrophilic filaments, just like in fascia. Lubrication of joints is another critical area that is now not so well understood, but I think that it's possible that the EZ water is critically involved in that, and injury-induced swelling as well can. So I can't guarantee that similar principles would play out in terms of fascia, but because we haven't studied it directly, but indirectly, it seems that the water is really important and is going to play a critical role. So this is discussed in the book that was mentioned. It's become rather popular. I like the cover, my son, the artist, and also other illustrations. And it's even the fourth phase has even made its way to my granddaughter. So I think I will stop there. And happy to answer questions. So thank you for listening, watching. And should I stop sharing?
Yes, yes, you would.
Before I do, I would like to, offer my own toast, and my toast is my toast is to the future of real science, which is a science that goes in whatever natural direction it happens to go. It doesn't need to follow the tradition or fall in line or with what everybody else thinks, because I think maybe it was Mark Twain or someone like that who once said, if everybody is, or maybe it was Benjamin Franklin, if everybody is thinking alike, then nobody is thinking. So to the future of science. Thank you.
Thank you, Gerald. So don't run away yet. We have.
I'm not running away.
For you. Perhaps if
Okay.
you could stop screen sharing.
Okay.
The green thing at the top.
Oh, the green thing. Mine is red. Stop share. Is that okay?
Yes, whatever.
Okay, got it.
Splendid. Splendid. And perhaps we can invite everyone to turn on their cameras and we come back into community mode. Neil,
Well, this might be a good time. You know, I can tell by Steve's body language that he has something he would like to say. So I'm going to ask Steve to go first.
Okay.
Let me see if I can you let me put me in view here. Speaker view, if that's me. And I'm going to share my screen if I can.
Oh, okay, hang on, Steve. You just turned. Can you turn your camera back on and share your screen? I think you're a co-host.
My camera's
I'm gonna
on. There I
spotlight
am.
you.
Can I share my screen for a second?
Can you? I think so.
Yeah, I'm sharing my screen. I
Great.
just thought I'd show Gerald that's my knee under load, under anesthesia, and we can never close that space.
Yes.
There
yes.
was always a space in that joint.
Yeah.
Under load.
Yeah, I've seen pictures like this. Maybe it came from you. I'm sorry.
Yeah, this is me.
Yeah. Okay, that is you.
Yeah.
Yeah, it's amazing that you put a load and the two surfaces just don't come together.
Yeah, there's the meniscus right there.
Yeah.
And I had a guy pushing as hard as he can. This was a 230-pound former Notre Dame football player.
Oh my goodness.
Who turned physician, who put his full weight against me. The anesthesiologist was holding my shoulders so I wouldn't be pushed off the table. And the scope never closed that space.
Fantastic.
So to confirm your talk, but this gives another reason for that space to always be there.
Yeah. Okay. Yeah. I hear you, and I've seen that evidence before. And in the case, if it's really that positively charged water that's doing it, with the protons repelling each other, the more you press, the larger the repulsive force, because the charges are getting closer together. And so in theory, you should never be able to close that gap.
Right. And this also fits in the Tensegrity model because it's a combination of structure and those forces that are doing
Yeah,
it.
absolutely. Yeah, thank you for that.
Well, that's wonderful. I'm so excited.
Me too.
Gerald, can I
get out of here?
Gerald, can I ask you a little about your experimental work? Have you done anything directly with collagen? I know Naphion is one of your favored materials because it is so well suited to these bench models. Have you done any work looking at the behavior of collagen in your lab?
Well, not specifically, no, but other groups have, and reported indeed that there is what they mentioned structured water. They didn't report EZ, but yes, absolutely, there are layers of some kind of crystalline-like water that are associated with the collagen. I'm sorry, I can't recall. Ivan Cameron is one of the researchers, and so if there's any interest, he's from Texas. Where in Texas?
And his first name is?
Ivan, I-V-A-N, Cameron,
Okay,
C-A-M-E-R-O-N.
Great. Okay, thank you very much. Well, we have one of our enthusiastic colleagues is Jean-Claude Gamberto, who is
Yes.
a hand and plastic surgeon in France. Unfortunately, he's busy with grandchildren today, or he would be with us taking part in this conversation. But we've looked at many of his videos of fibers as they move within the body, and they all glisten beautifully, almost like icicles. And one just has an inherent sense that these fibers are all coated in water. Have you had opportunity to study any of these phenomena?
Well,
can I jump in? I just want to say,
Yeah.
so thanks, Neil. I was in touch with Jean-Claude Gamberto earlier, and he thinks that he might be able to jump in after the grandkids are popped into bed. So he might be jumping in a little while. So we'll keep an eye out.
Okay.
Yeah. Okay. In answer to your question, we haven't studied that directly. You know, I only wish in Seattle the days have 24 hours. I'm not sure where other places, and there's only so much that we can do, you know, with limited resources, limited space, and whatever. And we would like to study all of these, but we simply are unable. And of course, the fascia, the glistening character of the fascia is suggestive that something crystalline or liquid crystalline exists on the surface of the fibers, and that's what easy water is exactly. It's a liquid crystal. So you know, and I've seen Jean-Claude's video, a couple of them, and it struck me also. And you can also see little droplets of water moving along, and I suspect the principles are the same as the kinds of principles that are driving flow through those tubes that we find. So yeah, a lot of similarity there, and I wish we did have the capacity to study that, and maybe in the future we will. Thank you.
I have a question here from Kevin. Do you have any thoughts on what causes the noise when you crack a joint if the bones that make up the joint are separated by easy water?
Yeah, I actually did. Let me think. It's been a while since I thought about it, but it was an issue that came up. A volume change, a quick volume change, is what you need to crack, and it's possible that. See if I have this right. You know, easy water is. I'm thinking out loud right now. Easy water is denser than ordinary water, and so if there's a transition between easy water and bulk water, this will give you effectively a change of volume. If you have a change of volume, you're going to get a noise that comes out of it, because you know, that's how you create sound. And so if you I'm trying to think in which direction this would go. I apologize, because I had it worked out many years ago, but nobody's ever asked that, and I've forgotten. But I think it probably lies in a conversion either from easy water to bulk water, or possibly the other, if you put pressure on it, we know that pressure builds EZ. We have experiments that that demonstrates that. And it makes sense because if you take bulk water and you compress it, you're driving the equilibrium toward the EZ water, which is denser. See, so nature wants to do that. And if you change the local pressure, which you do by pushing on your knuckles, there could be a transition from one to the other. And I guess that's the route I would take in trying to figure out why you get that crack. Is a transition.
I also have a question here from one of our great model makers, Gerald de Jong.
Excuse me, Neil. Excuse me, Neil. Can I just interrupt a moment to say we're an hour in now, and I wonder if everybody could turn on their videos, or perhaps the tech team could work out how the videos could be turned on, and we carry on. Sorry about that, Neil. Carry on.
Not at all. I have a question here from Gerald, who asks, could easy water somehow represent a compressive force relative to nearby tensional force?
Can easy represent a force? I'm not sure what's meant by represent a force.
Could it perhaps could it constitute a force?
Well, I mean, easy as a material. You might say liquid crystal. By the way, side point, a group in Italy has been able to obtain now easy water, solid easy water at room temperature, solid water at room temperature. And we've confirmed it now, putting together a paper on that. So it's a substance of a sort. We like to think of it as a liquid crystal. And as such, it can provide a resistive force. If you push against it, it's going to resist because it's a lattice. It's not individual molecules that are bouncing around a gazillion times a second. It's like a semi-solid. And if you take a semi-solid and you exert force, it's going to resist. So it sustains or it resists compression. And it also, in some sense, it could provide an expansive force because as it builds, it pushes everything away, everything in its path. And so yeah, I mean, forces are intimately tied to the EZ. I wouldn't say the EZ is a force, but it's a material that can either provide or sustain forces. I hope that answers.
And several other questions, both from Chris and Susan, ask whether the forces of EZ water relate to or contribute to tensegrity architecture on a molecular scale.
Well, it's possible. You know, I mean, if you, for example, if you try to compress the EZ water, it's going to expand in the opposite direction. I mean, this is one maybe one aspect. It's not going to expand too much because those hexagons will like to remain as they are. It's possible also, you know, at the level of at the molecular level where you have these hexagons. You know, if you try to compress a hexagon, the layers are building like this, and if you look down, you see all the hexagons, right? And so if you were to pull in this direction, should compress in this direction, I would presume. So, you know, I actually, if you think about gels, that's another example where tensegrity could be. And you know, inside the gels, we know hydrogels are filled with EZ water, and if you take a gel and you squeeze it, it's going to expand in this direction. So I'm not sure, but it's possible that some of these features are translatable or relatable in some way to tensegrity. But I claim no expertise in this area, so I may be diverging off into Netherland by suggesting such.
I have a question from Carol Davis. Is it true that H3O2 binds to the double helix of collagen? And do you suspect that this phenomenon may contribute to the pathways known as fascial meridians in traditional Chinese medicine?
Well, yeah. In traditional Chinese medicine, by the way, I just had a healing yesterday, a remote healing from one of the more famous Chinese healers. I happened to strain my back a couple of days ago, and we were discussing some collaboration, and he took care of me. I was from quite a distance. I think he lives in Toronto right now, and you know, within 10 minutes, my back pain practically vanished. So it was really impressive. So yeah, the meridians. There's a group, I think it's a Korean group, maybe, who studied and came to the conclusion that the meridians may actually correspond to regions of EZ, water that run through the body and taking certain routes. And I'm not sure because we haven't studied that, but I certainly can imagine if you have EZ, there are so many things that are associated with the EZ. Certain kinds of energies that can run through it. You know, so the charges. I haven't had an opportunity to present this here today, but if you have a sort of a cable of EZ material and you add charge at one end, the charge is likely to transfer, and the EZ could retain either more or less charge. And so if it's really true that the meridians correspond to areas of EZ water, it would be easy for them to transmit charge and therefore transmit information along them. So it's an area that I think hasn't been studied, but needs to be studied because, you know, there's certainly a link between ancient Chinese medicine and water. Water has stood at the center of medicine and philosophy, not only the Chinese tradition, but the Ayurvedic tradition as well. So yeah, it's somehow related, and I think it's time for people to begin studying this. Yeah.
And then another question now from Diane Woodruff. We're told that overstretching wrings the water out of the fascia and weakens the body. Do I understand that the structure you've shown us is not broken by the stretching, but the hydration returns and the limb is restored?
Well, I didn't get the last 5 words. I heard you, but I didn't understand. But I can say yes. I mean, imagine a fascial entity, and you've got EZ water, and the reason the EZ water sticks to the protein is that the charges on the protein match the charges on the hexagons. And so you could get the first layer, and then the second layer sticks to the first, and so on, and the EZ builds. Now, if you were to stretch it, you no longer have conformation between the charges on the fascial material and the EZ water. So the first tendency would be to break some of those bonds in the EZ water, and I suspect that's what happens, and so you can get a degradation thereby of the EZ water by stretching. I mean, that's a simple answer to what could be a complicated question. it makes sense to me. I mean, you take a structure and you pull it and it tears.
Fabiana Silva asks, is the water bound, and is it also easy water in the extracellular matrix?
Yeah, extracellular matrix almost certainly has easy water because the proteins are mostly have surfaces that are hydrophilic, and our experience is that any biological surface that's hydrophilic will have easy water associated with it. So, I know this causes a bit of confusion because we think of the inside of the cell as being negative and the outside as being neutral or positive, and that stems from lots of experiments that were carried out using isolated cells in a bath. So we'd like to assume that in the body, the cell is similar to what we can measure in vitro by putting a muscle cell or a nerve cell in a bath of saline. But it's not, because outside the cell in vivo is a matrix, and so it's different. And so the common concept that we've got negative inside the cell and positive outside the cell is questionable, because the architecture is so different inside the body. This is a point of confusion, I think. But the evidence is suggestive that, yeah, you've got an extracellular matrix, and since the matrix consists of hydrophilic proteins with hydrophilic, or glycoproteins with hydrophilic surfaces, there should be easy water out there too. It's not so easy to test, because putting electrodes in you or me, fine electrodes that get into the cell or outside the cell, I think the technology is waiting to be developed to do that properly. I think once it's developed, there'll be some surprises.
Wilbur Kelsick asks about glycosaminoglycans. He says, how does easy water relate to the GAG in the matrix?
Well, it should relate rather directly, because these are hydrophilic materials, charged, highly charged, and therefore they should build easy water. Now, I should say that not every hydrophilic surface builds easy water. We've tested some that apparently don't, but most of them that we've looked at do. And every biological surface that we've looked at produces easy water. So I think, it may be that biology is by design, biological materials are biological because they interface with easy water. And, easy water may be rather fundamental. I know that Luc Montagnier who studies water now, and structured water and information in water. As you probably know, he won a Nobel Prize for discovering HIV. And he's been suggesting, he has evidence that the water that surrounds the DNA is easy or structured or easy water and can contain information. You see, that's the more interesting part. And that information can be transmitted non-chemically, somehow by electromagnetic waves to water. It's so interesting, and it may be that water and biological molecules are so closely tied to one another that he's suggesting that the water came before the DNA. And we like to think that there's DNA and cells built based on the DNA. And he's arguing, it makes sense to me too, that the water came before the DNA, and that nature, in order to preserve this kind of structural information, nature built DNA, RNA and such, but the water actually came first, the water containing the information. And, easy is just right for containing information because in some sense, it's like a computer memory. Computer memory consists of three-dimensional array of transistors, and each one is regularly positioned in this three-dimensional array, and each entity in that array can have two states. We say 0 or 1 or on and off, and that's how the information is stored. And the EZ structure is very similar. It's a three-dimensional array of oxygens and hydrogens. And if either the hydrogen or the oxygen could occupy two different states, then you've got the basis for memory. And a lot of research has shown that EZ, or I should say that water, and I think it's EZ water, can contain information and transmit information. And it turns out that the oxygen molecule can occupy not two states, but 5 different states. These are oxidation states. It could be -2, that's the common one, -1, 0, even +1, and +2, they're less common. The fact that oxygen it comes from ordinary chemistry, the book's nothing exotic. The fact that the oxygens can take on 5 different states means that the EZ array is not only capable of storing information, but has a capability far, far greater than standard thumb drives or what have you, standard memories. And a possibility in the future is that your memory in your computer will be replaced by EZ water, especially EZ water, which at room temperature can be a solid. I would make that as a suggested prediction for the future, because the memory density in theory, we don't know how it works yet exactly, but in theory, it could contain so much capacity for information. And a lot of that may be in the fascia too, with the EZ water intimately involved with the fascia. So I'm sorry, long answer to a short question.
No, that was very good. Let me suggest that we'll take one more question from the chat, and then perhaps we'll open up and have a more free-flowing forum. Susan asks, as you study flow through the tube, is there any indication as to whether it flows helically?
We can't determine that. We don't have the resolution to determine it. We look at the flow through a microscope, and the way we can detect the flow is we put some markers, microspheres in the water, and we could, using a microscope, we can track the motion of those spheres along with the water. And we don't have enough resolution to test to see whether that flow is helical. I wonder why this comes the suggestion that it might be helical. I mean, it certainly might, but it isn't. I'm just wondering what is the basis for that question. But I guess it's not my role to ask questions.
Can I ask you to
I'll just say thank you. When I turn on the tap and I watch the water come out, it flows helically. When you pull the plug on the drain, the water doesn't follow a straight line. It flows helically. We think of efficiency as being a straight line between 2 points, but sometimes obviously efficiency is helical, and there's a lot of evidence that blood flows through the veins helically. So my curiosity comes from that.
Oh, okay. Yeah, well, I mean, you could be right on, but we just don't have the evidence. So I'm sorry.
No, that's fine. That's great.
Yeah, okay.
Can I also say, Neil, when we're opening things up, we do have a lot of questions in the queue, and I'm wondering if we should sort of ask those people who have put their questions in the chat to go, but whose questions have not been heard.
I think Gerald and I are both getting a little exhausted, so let us pass the parcel to you, Susan, and see if you'd like to be the director of operations.
All right, I'll just go down the list that they came in the chat, some of which came to everyone and some of which came to me directly. Gregory Chudy, you had a question about, isn't it really just that we have found another way to get energy from light? Gregory, do you want to ask that? The way you were talking about it? You have to unmute. He can't unmute. Are people allowed to unmute? Mariana's taking care of that. Thank
There we
you.
go. My question was mainly just plants photosynthesize. Do we have a term for our consumption of light, whether it be similar or very different than how plants do it?
Well, yeah, at first you'd think it must be different because the plants are special. But I came to realize that why should it be different? Why should nature do it in a different way? It just turns out that it's not only the chloroplasts that are responsible, the so-called antennas that get the light, and step one in the photosynthetic process is the splitting of water. That's been known for a long time, and it's 100% efficient. The other steps that follow in photosynthesis, I think there are about 20, are not well understood. And it's possible, this is a speculation, it's possible that the mechanisms of photosynthesis and the mechanism that we're describing are exactly the same. It may be that photosynthesis is you might say, like a special example of a more generic process. And with the first step being light causing a splitting of the water molecule. It's also possible that in the 20 odd steps of photosynthesis, one of the reasons that is still unclear how they work is that the photosynthesis people don't understand all of what we've found in the past years about what happens when water splits. See, they don't have that on their map, and so if you start with a foundation that is wobbly, it's very difficult to build a structure on it. The structure is going to fall apart without all kinds of struts and braces and whatever. I don't know that that's the case, but it's possible that photosynthesis is simply a special case with very high efficiency of what we've discovered about what happens to water when light hits the water. I'm not sure of that. I would like to speculate that, but it remains to be seen.
That's great. Thank you. Steve, do you want to unmute because you had a question about oxidicity?
It wasn't a question. I was just guessing that the physical response of structured water may be oxidic instead of linear, and that it expands and contracts oxidically. And it's easy to demonstrate that muscles contract and when the so-called contraction are really expanding oxidically. And I would just think that the structured water would fit into that model.
Yeah, I can't. Well, muscle certainly is certainly true that when the muscle shortens, it gets fatter. But beyond that,
but they don't have to shorten to get fatter. You can get the muscles to get fatter without shortening.
You mean at the molecular level?
No, at the organism level. If you grab a hold of something hard in your hand,
yeah,
and feel your forearm muscle and squeeze,
yeah,
without shrinking your hand at all, the muscles in your forearm all expand. They don't, and there's no shortening.
Well, okay, I hear you.
Can I jump in for just a moment on that note?
George, okay, but I do have your question in the queue,
Okay.
George, but if this
Well,
is
I just wanted to add to this part about the oxidic nature, only because, your models, Gerald, thanks very much. It's a pleasure to meet you, by the way.
Thank you.
Your model is looking at a linear surface, hydrophilic surface, but you have to remember in tissue, we're bound in structures that are bound in, various fascial elements that are, not linear. They're circular or spherical or ovoid in nature. So that hydrophilic surface is not a linear surface. It's a enclosed surface within, the myofascial, the sarcomere, all of those things. So you think of it as a linear surface over a spherical or an enclosed structure. So that entire expansion could account for what you're saying, Steve. Can you follow?
Well, it's you could say that, but I'm just, I'm doing a fact. If you hold something in your hand,
sure,
and feel your muscles squeeze without shortening any of your forearm muscles, those muscles expand and tighten. You can feel it.
Okay, can I respond to that one? When a muscle is activated, which is in your case, what we found is that the water, in the relaxed state, the water is easy water. In the activated state, it transitions to ordinary water. But the ordinary water has a higher volume. It's less dense by 10% or something like this. And so when you activate the muscle, even without shortening, if the water is going from easy water to bulk water, the muscle will expand. And I think this actually is rather nice evidence that I hadn't come to realize. This is consistent with the idea that the activation of the muscle is associated with the transition of the water from easy to bulk water, which is what we found. But I didn't think about the density change. But I think you pinpointed an important point.
Excellent.
We have Anna Atsubo next, and then after Anna, John Shorkey. Anna, you want to unmute?
Yeah, hi Gerald, thank you for your lecture, brilliant lecture. I just wonder, how does easy water relates to vibration or oscillation?
Maybe you could amplify the question, because I'm not sure what you mean by that. Could you say a few more words about?
Yeah, like suppose in physical therapy we use lots of vibration tools. I was wondering, how does easy water relates to that?
You people are asking penetrating questions. Subjects I haven't thought about.
Well, feel free to speculate. We understand the difference between research and speculation. So.
Well, I'm not sure if my speculations are worth any more than anybody else's speculations, but I'm trying to think of how vibration, so mechanical vibration, I think is what you're talking about. We haven't studied it.
But Jerry, when you think of it, right, everything's already vibrating anyway.
Yeah, well, some things more than others. So, if the easy water is actually a liquid crystalline lattice, it's not vibrating very much. It's not, things that vibrate a lot generate infrared energy. And the more they vibrate, the more infrared, and we interpret that as heat or temperature. And in the case of the EZ, if you were to use an infrared camera, which we did, and measure the effective temperature of the EZ, it's very cold, which means it's not vibrating very much. And that makes sense, because it's a lattice that's all tied together. It's not individual water molecules. And when individual water molecules bounce around fiercely at a gazillion times a second, there's a lot of infrared energy that comes from that. And we would say its temperature is higher than something else that's not vibrating. And the EZ doesn't vibrate very much according to what we measure. So I don't know if that answers your question.
Yes,
thank you.
We do have a lot of questions. So let's go to John Sharkey next, and then after him is George Roth. John, are you able to unmute?
Sure, I thought you were going to read my question. I just wanted to ask, Dr. Pollack, by the way, Dr. Pollack.
John, it's more fun when you get to ask, right?
All right, yeah, for sure. I think we were supposed to do a little presentation together for Dr. Carla McMakin, but unfortunately, because of the COVID, we won't get to sit down. I won't get to sit down with you and have a beer. And
well, you're not missing too much, but
I have a million questions for you in relation to circulation and so on. That's my problem right now, because from a circulatory viewpoint, the whole thing is crazy. The whole thing is absolutely crazy. Even the position of the heart within the thoracic cavity, if it's a pump, that is, if it's a pump. Why would you put it there as opposed to down at the feet, for example? The fact that blood comes to almost a standstill, quiescence, it almost stops in the capillary beds. And yet by the time it comes back to the heart via the superior inferior vena cava, it's actually traveling at the same speed as the blood that's leaving through the aorta. So I think your hypothesis is one that certainly resonates with me. And I'm writing a paper on that particular topic just at the moment. But I don't know if this is too vague a question to ask you, but is there a relation, or how does your model relate to stigmergy, or does stigmergy have an impact?
Well, I don't know that word. Could you explain?
Okay.
Sorry.
No, I think it'll be too complex for me to. That's a question then for over a beer.
Okay, over a beer. Who treats, me or you?
Oh, for sure, me. Are you joking?
Oh, that's great. Okay, looking forward.
Great. Okay, so we'll go to George Roth and next. Thanks.
Oh, hi again. So I might have missed this, but you're a student who came up with the stimulation of the water using light. Are you stating that light is the main only driving force? Because I'm thinking of deep sea creatures, creatures that live in deep underwater caves that have never been exposed to light. They are generating physiological function without the use of light. So other forms of energy besides photonic stimulation, I'm thinking biomagnetism, ATP from food sources, or is that something that you also relate to the generation?
Oh, well, we haven't studied everything, and so we don't know what might contribute to the buildup of EZ. Obviously, we can't rule out something we've never measured, like cosmic energy, for example. But as far as the creatures at the bottom of the sea who've never been exposed to light, actually they have, because of the deep sea thermal vents that are generating oodles of infrared energy because the warmth down there. And so so you could call it light. The physicists will call the whole spectrum of electromagnetic energy light. Of course, the visible range, what we see, is only a small section of it, but it's a huge expanse. So we use the term light in a more generic term that contains all the wavelengths, or many of the wavelengths in the spectrum, not just the ones we see. So yeah, it's dark down there. If you were to immerse yourself way down deep, it's dark. Your eyes can't pick up anything because they're sensitive only to the visible wavelengths. But there are lots of other wavelengths, especially the infrared, that will provide energy for them to thrive. And it's really the infrared that makes the difference. That's what our experiments show.
What's interesting about that, if I can just chime in for a second, is my daughter studies biology, and one of her friends, I said, well, how did those things so down deep, so far away from the light, how did they ever come to be? And he said, I think that's probably where it started, that they were starting with that energy. Life was starting with that energy. And then as it came up closer to the surface, it added in this ability to take in light. And so that just blew me away, because it flipped everything. But what you're saying, Jerry, makes complete sense.
Well, thank you. I think it makes sense too, and you know that life originated in the sea, plenty of energy, although not the kind we think of.
Yeah, I guess I just to add to that. Then you know the stuff that I've been looking at. I'm just wondering if you'd ever considered, or maybe want to consider, applying a magnetic field to the water and see how that affects.
We already did an experiment, not published yet because the experiments are ongoing. So we put a magnet inside the water, and next to the South Pole and next to the North Pole, EZs form. You know, we put microspheres in. We could see the exclusion of microspheres next to both of those surfaces. The reason for that, I have my own speculation, and it has to do with the nature of magnetic fields, but that's beyond, I think, what the scope of what we're discussing. I think magnetism is different from what other people think, but yes, we studied it, and next to those two poles, EZs form, roughly one millimeter or so, pretty sizable exclusion zones.
Okay.
And
Yeah.
and next we have Wilbur Kelsick, and after him, Anastasi.
Okay, and I got to say that in about 15 minutes, I need to sign off. I'm sorry.
No, that's fine. Thank you for
Yeah.
staying with us. We'll get to as many people as we can. If you all keep your questions brief, then we'll get to more of them. Thanks.
Got to keep my answers brief.
Dr. Pollack, thank you for your presentation. It's very inspiring. I remember when you were in Vancouver in 2015, I think I asked you the question, but still wasn't able to get a sort of clarity around the idea that, so I am like you in the same school of believing that water is very important and it's hydrophilic and it bonds to hydrophilic surfaces and everything. And we know collagen is one of the substances that what we are made up of, that water has a higher affiliation. And it's my mind, in my thinking of the way of looking at the biology of collagen, is that collagen will not adapt to load efficiently. That's my hypothesis, without the presence of water. Can you comment on that, or is that a...
Yeah, well, you know, it's a sort of philosophical point that we like to think of collagen as independent of water, but the surface of collagen is normally embedded or surrounded by EZ water, and so the real molecule is what we call the protein plus the water. You can't separate the two because otherwise the protein is not in its normal state. I'm not sure beyond that how to answer the question, except to say that the two are actually merged together as one. And you know, the people who were studying protein folding, many of them failed to take it, or most of them failed to take into account the water and the fact that the water is stuck to the surface of the protein. And I fear that, you know, if you don't do that, you get the wrong answer. And I think that's why the field of protein folding is, I think the field could advance itself much further if they took into account the actual structure in nature, which is surrounded by the water, the EZ water.
All right, I'm gonna push forward. Anastasia, are you unmuted?
Yes, I am.
Great.
Thank you, Gerald, for an amazing talk. I've been a fan of your books. Simple thought regarding lipid bilayers and their similarity to structured water and the whole idea of the polarity of structured water. And I loved the answer to, well, the idea that when you put a magnet in there, you got structured water around the poles. So I'm just curious about in biology, how biology is meeting chemistry here. Do you think that water became structured by forces before we got to the stage of developing membranes?
Yeah,
because it seems a similar process.
I do. I think it
in chemistry.
It makes sense philosophically. You know, you started with something simple and pervasive like water.
I'm sorry, I'm frozen, I think.
Yeah, your face is, your image is frozen, but not your voice. it's melted. Yeah, I think, you know, more and more now that it's clear that water can store, or easy water, I think, can store information. It's conceivable that things started with water, you know, on a primitive scale, and gradually nature started building proteins that interface with that water. And, you know, proteins could be you might say, a more permanent structure to store information, but they came later. So yeah, I'm with you on that. I agree with that.
Let's Jerry, how much more time do you have?
Well, I've got a 12 o'clock meeting, and so if I could get out of here at 10 or 5 to 12, that would be great.
Well, maybe what we should do is Chris has a question, and then maybe after Chris's question, we should maybe do our sponsors and close our formal YouTube session, and then move on. But we also have a YouTube question, so maybe we can get both of those in. Chris, you want to go?
Thank you. Yes, and thank you so much for the presentation. I've got more than one question. I'm going to try and zero in on one, and it's a question that Carol shares as well. And that is, in the Biotensegrity community, Steve's been bringing us to this concept of phase change and soft matter. And when I see your presentation and you talk about the easy water, to me, it's a phase change.
Yes, it is.
And I wonder how far you've explored that. And maybe after we've closed the show, I would love to hear Steve's response as well.
Well, yeah, I mean, what we're talking about is exactly that. It's a phase change. So, the cell, when a cell becomes or transitions between the relaxed state and the activated state, where it's doing what it's designed to do, it's a change of water and a change in the proteins. Essentially, everything in the cell changes from one state to another state, and that is basically a phase change. And the idea of phase change is not a new one, obviously, and it's been discussed, and I think now in the field of secretion, it's pretty well accepted that that secretion is a phase change that occurs. I mean, molecules are secreted by a phase change that occurs inside the cell or inside the secretory vesicles. That's just one example, but I think.
And,
go ahead.
And yes, for us, we have a lot of body workers here and a lot of movement practitioners. And so it would be super interesting to really also bring that into the experience of relief that people have when they have these sessions. And it does feel like the body gets less stuck or less gluey. But,
yeah, well, yeah. So the idea of phase change is in my 2001 book, Cells, Gels, and the Engines of Life. That's the central message in that book, is that there's a phase change that occurs, and that phase change occurs in many cells, and possibly all cells, whenever they go from the quiescent state to the activated state. A phase change occurs in the entire cell. So yeah, we're exemplars of that. Our body cells undergo that kind of change for sure.
Thank you. And for using the language that we use is helpful for me. Go ahead, Susan.
Oh, I was just going to say, it looks like the comment from YouTube is also a phase change. It's more of a comment than a question from Caden Rolfs. Thanks, Caden, about how joint fluid, something like oil becomes thinner with forces being applied, but joint fluid becomes thicker. So it's another phase change.
Yeah. Okay. Yeah.
Viscosity question, but maybe we've already covered it, or what do you think? Steve?
Covered.
All right. Let's do our sponsors, and then we'll sign off of YouTube, and I'll go back to the list of questions for those who are still with us on Zoom. And Thank you, Neil. I'm going to turn it over to you first, because I know you want to do a special thank you to Jerry, and then we'll go to sponsors and say goodbye.
Yes, Gerald, thank you very much indeed for the time you've given us today. We're all a little closer to understanding your ideas, having heard you articulate them so well today. I'm sorry if you felt that we didn't give you quite long enough to give the full range of the topic, but one of the ways to solve that problem may be to have you come back again after we have reflected and fermented some of these ideas. But thank you very much indeed. We're most grateful to you for being part of this conversation.
It's a delight. It's so gratifying to speak before an audience of open-minded people like your group. It's really a great pleasure. Thank you all. And may I now sign off, or should I be hanging in there?
Sign off, and can you sign off with a toast?
Yes. Okay. I will toast the open-mindedness of this group. A group obviously interested in finding truth and digging deeply in order to do that. It's a great pleasure to be amongst your group. There should be vodka in here, but I'm afraid it's just the spring water, and you understand why. So cheers.
Cheers.
Okay, take care, people. I'm
Jerry.
Yeah.
Thank you so much.
My pleasure. Yeah.
Thank you. And then who's doing our sponsors?
Chris, are you open to that, or?
Oh, I'm not quite ready. You go for it.
Okay.
Please. Sorry, Graham.
That's all right. Our Biotensegrity Party is an all-volunteer production of the Stephen M. Levin Biotensegrity Archive, and made possible through the generous support of our sponsors. Handspring Publishing have a wide range of authors and books available for movement and manual therapy professionals, and also produce a regular newsletter and ongoing webinar series with Elizabeth Larkham. Embodied Biotensegrity, who provide an excellent resource for those of you looking to learn more about biotensegrity. Artifact Pro in Madrid, Spain, who produce wonderful tensegrity models. Integrated Biotensegrity, with its upcoming 3-day movement-focused event. I don't have a date here, but perhaps you'll put it in the chat. Keynote speakers John Sharkey, Joanne Avison, and Wilbur Kelsick, and other international presenters. And finally, thefasciahub.com, who are running a free webinar on February the 24th with Roberto Ciaf, London-based professor with an international reputation in the fields of neurology, motor neuron dysfunction, and Guillain-Barré syndrome. So look at the chat if you can, support our sponsors.
And the International Congress in Biotensegrity that Anna Altuvo is going to be presenting April 23rd, 24th, 25th. Do I have the dates right, Anna? Yeah, but we don't have a website yet, right? Okay, we'll let you know next time.
And finally, we need to give an indication of what's happening next week.
Oh, next week, Eleanor Ori, Dr. Eleanor Ori is a researcher at the University of Maryland, and she studies microtentacles that emerge from cancer cells with changes in the tensile properties of the cells. And she's the one who came up with a way to measure the development of these microtentacles. The microtentacles make it easier, as I understand it, for the cancer cells to hitch a ride on the blood and promote metastasis. So she's going to be talking about her work. She's also an aerialist, a trapeze artist, and a contortionist. And she's so happy because this is her work with understanding biotensegrity is one of the first times that she's been able to connect her movement practices with her cellular work.
Great.
Great.
Thank you,
So
Susan.
before I sign off YouTube, I wonder if we can hear from Steve, also Carol, if it's okay to hear from Carol, but let's hear from Steve first.
About what? I mean, he's amazing. I mean, you have to know, but what happened during this talk is that a lot of things we talked about at the macro scale, he confirmed at the micro scale at the water level.
Hey,
So
Steve,
that,
tell everybody how when you two first met. I think it's great.
you mean online?
Yeah.
Yeah, well, I was involved in an embryology course online with Dick Gordon, who's a multifaceted genius, and he was running. He's an expert in embryology. He developed partly developed the CAT scan. He's a world-known person in diatoms and several other things. Anyway, he was running an embryology course that I was studied with him, and Gerald came aboard for a lecture then, and that's when I heard him first talk. I'd already read a lot of his stuff, but it's when I first heard him talk. So I interfaced with him there. But what he said was confirm the fact that muscles don't contract, they phase change. I mean, that was a very important concept, that we get a phase change with an auxetic expansion of muscle. They don't contract, and the odds are very high that when you put them in a closed kinematic chain, that they operate isometrically, so they don't change length. They just expand auxetically by a phase change. So when I've gone through this phase change over and over again with these things that go from one to the other, it's either one or the other, and it goes flips back and forth. And most of the things in the body are doing that. And we should look for that phase change where there's a sudden change from one phase to another. That changes the whole concept of what we're doing. So when you take anything out of the system, it no longer can be understood. You take a muscle out of the body and you put it in these testing machines, and it lengthens and shortens and all that sort of thing. When in the life situation, it probably doesn't do any shortening at all. It readjusts and it tightens, it tenses. So you get a change in the tension of the system, not a change in the length of the muscle. That's one thing, but he covered so many other things, but he reconfirmed what we said. And what particularly, of course, was the knee. I was so excited about the knee was because he showed us another reason why that space exists, but he confirmed that that space has to exist because you can't get the two south poles of a magnet together. You just can't do it. They'll just keep resisting more and more. And I've been saying there is a space there, and he confirmed that there is a reason for the space there besides the model of the Tensegrity, but of course it's probably their overlay one on another. And again, he did also the hexagonal close packing, which we've said. And if you take those hexagonals and put them in 3 dimensions, one on top of the other, and shift them slightly, you'll end up getting a Tensegrity cross-changing depth-wise, not just plane-wise. So they're not just sheets.
Phase change.
There's also depth to them, and they phase change, and they build a Tensegrity. So they're a Tensegrity structure. I can assure you that structured water, his easy water, probably organizes as a Tensegrity.
Like in the cyclohexane.
Right.
Yeah.
So it's, everything fits.
That's great.
Can
Carol?
I add something or no? Or am I muted?
Yeah, well, we were asking Carol to go next. Do you want to go before Carol, John?
God, no, never before Carol. No.
But that phase change, Steve, Jerry also has research about with solid bits of water at room temperature. So that is also like the cyclohexane at the triple point.
Yeah, it's all part of the same thing we've been talking about at many levels. They all fit together.
Yeah, there's so much that he touched on that we've touched on here that we could just join up all the messages. I'm looking now at an article by Mei-Wan Ho, Coherent Energy, Liquid Crystallinity and Acupuncture, where she talks about bound water corresponding to the more loosely structured water cylinder on the surface of the triple helix of the collagen, and so-called free water filling the spaces between the fibers and in between, and about the memory associated with that, the major factor contributing to the efficiency of intercommunication in fascia is the structured oriented nature of collagen, liquid crystalline fibers, and water having memory and so forth. But what I wanted to ask him again was people just coming into fascia for the first time very often say, why do I feel stiff? Where is that stiffness coming from? Is it hypertonicity, or why do sometimes I feel stiff and sometimes I feel so loose? And to me, it all comes down to this whole concept of what's happening with the glycosaminoglycans, Wilbur, and your concept of the gags and the water, and what's happening with the water in the mucopolysaccharide ground substance. And so when I asked Pollack at the Fascia Research Congress in Vancouver, I said, after his presentation, I said, those of us who do Barnes myofascial release say, well, when we place our hands in 60 to 90 seconds, our hands heat up, and then very subtly we can feel our hands kind of being pulled down, melting down into the tissue, and then there's a kind of a flow that takes off. And Barnes refers to that as melting, that the condensation of the ground substance of the fascia is going through thixotrophy, or changing form. It's gel, it's more solid going to more fluid gel. And Pollack responded, you're absolutely right. That's exactly what's happened. We call it thixotrophy. And it's through pressure and the infrared energy from your hands that's changing the structure of the molecule, so that then the fascia opens up. And then Leon Chetow later on in one of my readings of him, he says, when you take your hands off, the fluid from the capillaries flows in and you get that marvelous feeling of being opened up and more fluid. Also happens after we're exercising and running and so forth, and moving our muscles and pulling that water out of the capillaries into the muscle. That's what I wanted to talk to him about, but we didn't have time. And I have to make a confession, sometimes we hold on to these theories and so we read something, or science says something to us, and we said, oh, that's what fits my experience. That's it. I'm going to hang on to that. And then science never stands still. It's always evolving and changing. And, there was a little part of me that didn't want to ask him that question, because I didn't want him to say, oh, no, Carol, that's old data. We've got much more. And I didn't want to hear that my theory was wrong. And, it's just so human of us, isn't it, to want to hold on. But the fact is that we have to keep asking the questions. And his toast was wonderful. We have to keep asking the questions that make the most sense out of our experience, out of our lived experience, as Jaap would say. So that's all I really wanted to say. Thanks for listening. And there's a whole lot of stuff. Mei-Wan Ho, the rainbow and the worm, about water, polarized water, and infrared energy, and how you can get photons out of sweet potatoes that never saw the light of day. The third generation of sweet potato in dark earth, you can still get photons in them, because infrared energy comes from so many different places, not just the sun. Okay.
Okay, over to you, Mr. Sharkey.
Oh, wow, Dr. Davis has just changed what I want to say. But because that's brilliant, great wisdom there. And the truth is, Carl, that if we, I think that Dr. Pollack was saying, I believe, or I think, and then he went, no, hang on a second, I have to change that to evidence suggests. Do you remember that, everybody? And that's because from a scientific perspective, that's the type of language that you have to use. But if you stop and think about it, Dr. Stephen Levin has been talking about things since the 70s, and research is only now starting to catch up. That's the reality. That's the reality of it. And I was a little bit concerned that I know we have a mixed group here, some people who've been, really earning their stripes in terms of tensegrity and biotensegrity. But just on the off chance, people are listening to Dr. Levin there and saying, what's he saying, for goodness sake? Look, I do this and my muscle contracts and he says it doesn't contract. What's he talking about? Of course it contracts. You have to go back to the word contract. Look at the etymology of the word, look at the history of it, and say, well, why do they use that word? And the word contract, I don't want to spoil your enjoyment of going off and doing a bit of surveying research yourself, but the word contract means to shrink. That's what it means. So nothing is shrinking. And as Dr. Levin says, perhaps nothing is shortening and nothing is lengthening. It's shape changing and phase changing. So please don't get scared off or put off when somebody says something that makes you say, what's he talking about? The issue is that we've been studying this for many years, and perhaps something trips off our tongue that might cause you a little bit of confusion. So I think we're all open to emails and communication, and if anybody has some issues, feel free to contact us. But that's the main point I wanted to make, that muscles don't contract.
Thank you. And Eileen, you've got questions about water and water having memory. I know that in at least one of the very easily accessible talks that Gerald Pollack gives on YouTube, that are up on YouTube, he talks about the research with the snowflakes and the responses to different kinds of even words and music and that sort of thing. And then water and memory. So you might want to look into those.
Thank you.
Chris, we're still live on YouTube, is that right?
That's right. I was just texting you. Shall we sign off from YouTube?
Yeah, bye-bye.
Okay, let's everyone say goodbye to our YouTube audience. We'll see you next week, hopefully. And if you want to join us on Zoom, maybe somebody can put the link for our newsletter into the YouTube channel.
Yes, in order to join us on Zoom, you need to be on the email list of the Stephen M. Levin Biotensegrity Archive.
Okay, bye.